Very warm welcome to all the participants who have joined us for the 47th session of our monthly series BISMiS Live. We have with us an excellent and amazing speaker with us, Professor Shuang-Jiang Liu, or SJ, from the Institute of Microbiology, Chinese Academy of Sciences. Professor Liu will speak on a dialogue between microbiome and bacterial taxonomy. SJ completed his PhD, and then he is currently a principal investigator at the Institute of Microbiology of Chinese Academy of Sciences, and also a professor of microbiology at University of Chinese Academy of Sciences at Beijing and at Shandong University, Qingdao, where he is currently residing. And he is also the convener of the sixth meeting of BISMIS. He is also the head of the gut microbiome branch of the Chinese Association of Biophysics. His research interest covers microbial diversity in natural environments and human gut, microbial degradation of and chemotaxis towards pollutants in environments, and the interaction of microbiomes with hosts. So, with that brief introduction, I welcome SJ and request him to start his presentation.
Thank you for the introduction, Mr. Chairman. I will speak about something about microbiome and also taxonomy. So I have a topic dialogue between this microbiome and taxonomy. I hope people who are listening do not misunderstand. I will not cover the how to classify microbiomes. This is not the meaning of the topic, the title. Actually, I should have a more precise title. Probably should call the microbial taxonomy and microbiome, yeah, something like that. I was thinking how I start my talk. So I will start here. I will start by introduce my research with microbiology. I was trained as a microbiology researcher. As a student, a PhD of my PhD education training and also postdoc training. I work in microbial cultivation, microbial physiology, metabolism, and genetics. Well, this is my interest. Um, After I get my PhD, I start my research in the Institute of Microbiology. Mainly, I was working with environments. So it's a field environmental microbiology. That was my first 20 years of my research career. And then I come to work with human health. Um, there I work on cultivation, also the physiology, also particularly how microbes interact with human physiology or human genetics and so on. So that's my recent 10 years. Those research make me to uh, work on, let's say, at the beginning, my research mostly focused on one organism, and later on, either environmental microbiology or human health, the microbiome. I work with a lot of organisms together at the same time. Yeah, that's my background. Uh, and I never say I'm a microbial taxonomist, but I did, and I do describe a lot of organisms. Uh, when I prepare this talk, I check my publications with IGSEM. My first paper is 2002, and most recent paper is this year. Um, so all those years, I counted is about 78 IGSM papers. That's related to taxonomy and description of new organisms. But beside those, in my lab, we described about 8,000, oh no, sorry, 800 new microorganisms. But only about 300 names was validated with the uh, AJSEM. That's background. And so there are quite a lot of microbes did not their names they do when we describe a new organism we do give a name, but those names cannot be validated. And this is I will start something with this. Well, if you nominate you describe and nominate a new microorganism, you need to, my understanding, at least do those works to know the morphology and also the genome and also the phylogeny, either based on 16 sRNA gene or a whole genome. Besides those, you also need to characterize physiological, biochemical, or chemotaxonomic or ecology features of these organisms. And then you need to give a name. You need a name and also need the taxonomy ranks and so on. Then probably you describe your organism with the name validated, published with IGSEM. That's my understanding. But this is not a problem for most studies, particularly if you're dealing with a couple of organisms, not too much, not too much. But the question is here, if you work with microbiomes, you often deal with hundred or thousand microorganisms together. Here, I give two example publications. They are published in Nature Biotechnology and in 2000s. You see each of those papers deal with more than 1,000, more than 1,000 people, and it's really hard if you do the uh, taxonomy or the characterization according to the slides I just showed you. All those things you do all together. That's a lot of work. You cannot do it just in a short time or not easily. Um, so for some people, if you are not familiar with microbiome, the concept, simply say microbiome is a assembly of bacteria, archaea, fungi, virus, and other microorganisms in a given specific bio or abiotic habitats. I'll further give you a couple of examples. Here you see this size. This is a publication in Nature Microbiology. It's 2016. This paper was rejected. The rejection is not because of the science, it's because of the ethical problem. I think they cannot provide ethical certificate or something like that. So the paper was extracted. But the paper, they described they have 247 new taxa. Still today, there is 117 remains. Their taxonomy of their names are not effectively nominated. So that's one example. So the name is not they are not effectively named the organisms. Another two example, also publication in Nature 2016, and the other one is 2019. They describe a lot of new organisms. In the Nature paper, they claimed they have 63 new taxa, and in Nature Biotech, the 2009 the paper, they claimed 105 new taxa. But if you check the sequence of those papers, I mean the microorganisms, the sequence of the microorganisms described in those papers, they have 54 taxa. Their sequence are almost identical. So that means they overlap. They are overlap. They repeated claim. One paper claim is new organism, and the other also claim those are again claim they are new organism. So I think this is not good. If you do not nominate or if you do not give a taxonomic status and a name, and those sequence or those microbes will be repeatedly regarded as novel by different studies. So I think taxonomy characterization of new microbial taxa after those large-scale cultivation, not only microbiome to large-scale cultivation, it's very important. But this is, I think, it's not it's neglected still right now. I served as AEM, Applied Environmental Microbiology, so as editor for 10 years, then quite some years ago. I did 2 terms of editor, and one of our editor sent me once. He sent me an email. He's dealing with manuscript on sponge microbial diversity, and he said he said like this. He read quite some papers, and the paper described they have a lot of new taxa, but then taxa later on some other study or even the same studies claimed again those that previously claimed as new taxa, again new taxa. So this is not a problem right now, but quite some time ago, I mean. Um, so give a name and also the taxonomy status is important for either microbiologists or people work with microbiome and so on. I think people probably know this, but why people do not give a name or do not do classification? Probably one thing is there is obstacles. There is difficulty in doing this work. On this left side, you see this is a chat I cited from the paper, from the 2010 paper. The summarized, if you describe a microorganism in different journals, including our IGSAM journal and other journals, it takes, you see, quite a long time, quite a long time. I don't know if the IGSAM, how long it takes for a paper. My experience probably not as long as this right now, probably 1 year sometime. It depends on the editor and the reviewers, of course. But still, I think quite a long time. Besides those work, you need to find the organism, you need to deposit them in different culture collection centers, and also you need to characterize the organism. So that's a lot of work. And those are researches and write a manuscript and submission and so on. So before that, you need to know the ranks, the entomology and so on. For publish new taxa and to validate by ICSP, you need to meet those standards, those requirements. Those are obligatory requirements, and so on. This is a lot of work, and I think it's important to do everything precise and important. But if you don't nominate and don't describe the organism, and you publish a lot of paper, and then you make those problem as I said, that's also not good. So in my lab, we proposed, at least we do this also in my in the lab. We proposed a simplified and we call the minimum requirements for taxonomy description. Thus, we run this in the lab, in my own lab. This includes four steps. If you find the organism, first you want to see the morphology. Well, as a trained as a microbiologist and my age, we use microscope. So I want to see what this microbe looks like. It's a rod or a coccus or so on. So first we will take a picture, electronic or light microscopy. We see the morphology, and nowadays it's easy to get a 16S or the whole genome and analyze the phylogeny and also the genome features. And then we do a phenotype, the physiology, what kind of carbon source, what kind of enzyme this organism produce, and the composition of cell wall or lip membrane. Well, this for phenotype, we don't have a certain, we just for example, you can use a biolog for carbon source assimilation, but if you don't use biolog, we just have a couple of sugars or other carbohydrates or short-chain fatty acids as a carbon source. This is you can select from your lab list. And then we deposit our culture. We deposit in our CCGMCC, the Chinese Culture Center for General Microbiology. Also, very important, we deposit one strain in our lab. So that's we make a biobank of we work on gut. We call it gut microbiome. And this example, what we really do in the lab, it's a flowchart of work, how to deal with microbiome, gut microbiome. I mean, get samples and cultivate all microorganisms and do 16. And if this organism is a new, and then we go this, we call it a simplified protocol or minimum requirements to the characterization. If not a new organism, and then we just deposit the culture in our culture bank. So this is the general flow chart, work flow chart in my lab. We use this flowchart, this protocol for mice microbial cultivation and characterization. So for this work, it's already published 2020. I think it was also the first systematic cultivation of mice or mouse gut microbes. We got about 1,800 assays. It covered 126 species. We also sequenced all those species. And also, there are 77 new species, and some are new genera. I think because the mice gut is not frequently used as a sample for cultivators, so there are a lot of new microbes. And then as a side, we published this paper 2020 in Nature Communication. Also, we provided the name and also genome, also some phenotypes. We also provided this with the publication, but this published as a supplementary material, And then later we learned that if you published as supplementary material, those cannot be used for validate your bacteria, your name. So all those bacteria name cannot be validated, although this bacteria is really, we think is uh, characterized to some details. So that's a lesson we learned. But right now, there are new taxa have been validated, published again by so people find those organisms and do some additional work and they publish the name. Then that's one example. To work with microbiome, you need to be careful. Do not include those in supplementary data. Second is also we work with human gut microbiome. Um, for this, I will make this short. We cultivated about 10,000 SLAs and we deposit more than 1,000 strains in culture collection centers. And this microbiome, this biobank contained covered 400 species, and there are 100 new taxa. And for this, this time we know we need to publish this the description of those strains in the main text, but that's quite long. So we made a table like this. Taxonomy, the name, the rank, and the etymology, and we need to designate type strings. Also a very short description of some features and the culture collection number and so on. Those are obligatory for validator name with HSAM. So that's those. But even, yeah, that's the description. Those, though with those, we validated, I think, half of them. Yeah, actually, 60 new taxa was the name of the 60 new taxa was validated in one time. We submitted all the materials, but still there's about more than half cannot be validated. The main problem is the culture collection accession numbers. We have one deposit in CCGMC, CGMC, but you know, gut microbes, sometimes it's not easy to be cultivated. It's easy to die, or if you transport your strain to another country, and then you need a lot of documents. Also, the time is not short. The transportation takes time. And some culture collection center cannot re, how say, cultivate those organisms again. When they arrive, probably they are died properly, or the cultivate condition is not meted. Uh, so the strain cannot be revived. Also, we also have problem to sending out some strains because they are some are potentially pathogenic, and so you cannot transport those. So that's a problem we met when you work with microbiome. Then that's something you need to be careful. Well, so at the beginning, we use this simplified workflow in my lab. I think right now it's really some other lab maybe can use this. The problem is if you nominate a new taxa, you need to do those, as I show in red. Those are not for many. I think people they are not familiar with those entomology and so on. You need to know Latin, and so you need to get help from some specialist. That's uh, one thing. And the last three slides I show, besides difficulties or those factors I described in my previous talk, also there are other potential pitfalls in claiming novel taxa. Those including some people working in the taxonomy, working with microbiomes. They use really a short 16S. They define a new taxa with short 16S. And some other people, they use previously published the taxonomy unit and they use the genome, but they did not recognize previously published genomes. And also there's repeats, as I said before. This because they use they use a data bank quite not updated data bank. So those are pitfalls if you nominate a new organism or if you claim a new bacterial you detected or you discovered. This is an example. This is a paper recently published, and I read this paper. I find, oh, this is I need to look into. The paper described a lot of new organisms, but they use the 16S. The 16S was extracted from draft genome. Normally, the 16S have about 1,500 base pairs, as shown in this line. But they're in their papers, they use a lot of short 16S, as you see by those the red dots in here. If you statically, you see this size. Most of their sequences are okay, but indeed there are quite some in this amount, 500, 1,300. There is more than 10 of they claimed new organisms. Their length is even short than 500 base pair. This is quite low quality. And that's I think a problem make those short 16 it's hard to describe to distinguish them from other sequence. I think other organisms. I mean, so they claim they are new. And also in this paper, the overlooked a lot of previously published names. You see here those 26 names are previous published, but they claim they are new clusters, those numbers. They claim those clusters are new organisms. Actually, those clusters are represented by those previously published organisms. So that's well, I think there's a solution to solve the problem. And also here, I summarize my talk. People should timely validate reported new taxa. This is important. This will avoid repeatedly claiming new organisms. Also, people should give enough credit to previous work and use updated data database, not on short sequences. And actually, this is what I really emphasize. We know there is a new name system, C code, And the C code use genomes as a standard. Well, I'm learning this C code right now. Uh, for nominate organisms. Uh, but I think only sequence is important, but probably it's not enough. I would like to see if you nominate, if you say, claim you find a new organ, you need to see it. Only sequence is not. Um but I see SASP, sorry. Uh, as I talked, the SCSP required quite a lot of work for description and nominate new organisms. Uh, in the future, as I learned this C code and CSP nomination, there is some difference. We probably need to coordinate in the future just for science. Also, I think it's easy for microbiologists, also for the people who work with microbiome. And really, I'm looking forward if we can discuss more in the next business meeting. Um, as the beginning, Mr. Chairman also uh, talked about this. So there's meeting in October this year in Qingdao, and so you are welcome, and you are invited to join the meeting. This meeting is quite, as we understand, not only in, nominate, there is broad interest topic and so on. It will be an interesting conference. This is my talk. I hope I make me clear is the taxonomy is if you find organism, particularly today you work with microbiome, often you meet a lot of organism. We should probably work carefully, describe new organisms. Also, this is from microbiome part. From taxonomy part, it's really difficult if you characterize each of those organisms according to the obligatory requirements by ICSP is really time consuming and labor costing. There should be a balance. That's my meaning. The title, the dialogue. We need dialogue. Find something in between. Make the people work with microbiome, particularly those cultivate and describing organisms, new organisms. Make them can do this. Do not leave the new what you claimed new organisms not named, not described, and then that's make a lot of confusion in the future. Okay, that's my talk. I would like to discuss more if there is questions or.
Thank you, SJ, for that very interesting talk and raising some key questions about, how naming of microorganisms is now um, being held up. We've received several questions, and Aaron had requested that he would like to make a few comments. So I have unmuted Aaron. Aaron, would you like to speak up?
Yes, with pleasure. You hear me?
Yes, yes, we do.
Yes.
Great. I first of all, I want to congratulate SJ or Shuang-Jiang for this wonderful talk. I was really looking for this. And I also want to congratulate him for A, describing, as he said, about 800 new taxa and succeeded in validating about 300 of them. It's quite an achievement. I know what it means, and what you have been doing and are still doing is just great work. I want to refer to a few things you said during your talk. One comment, I was not very happy to hear this, but you may be right, that when you submit a paper to IGSCM, it can take even more than a year until it's published. I am very disappointed about this. I was editor-in-chief of IGSCM for many years. I'm no longer involved in the editorial process, so I cannot be blamed here, but I hope that some of my fellow editors who are in charge of this now are listening to this and will try to improve, for there is no real excuse in my opinion. Then you raised the issue, and that is correct, of course, about supplementary material to publish protologs of organisms. Indeed, Rule 25A of the ICNP of the Proprietary Code states that supplementary material is not or no longer allowed as effective publication. This is a matter of what is a permanent record of publication. The ICSP has voted on this. So I am sorry that you were confronted with the problem, which you learned probably too late, that supplementary material is not accessible. But then you stated, and this is very good, that these tables that you present are perfectly okay for effective publication. Presenting the protologs in a table form is simple. It doesn't take much journal space. Journals will not raise problems when you do this. And something that's also important if you say things are it takes a long time and much work to characterize all those organisms. The ICNP, the code states, Rule 27, that the protolog must present a description of the properties of the taxon, but it does not state which properties. So the code does not tell you you have to do this and this and this and this and the long list of requirements, just properties and which properties are essential. This is not a restriction of the code. This is more a restriction of editors who think that certain properties are important for a certain group of organisms, etc. So in this case, the restrictions of the code are in fact not restrictions. If I understand well, the bottleneck of how many of these descriptions you can handle and get validly published is the requirement to deposit your strains in two culture collections in different countries. And here I and my colleagues cannot assist you here. This is a restriction that the ICSP entered in the code I think in Sydney 1999. And I think it makes much sense to have type strains deposited and available from culture collections in more than one country. The other bottleneck that you stated, at least potential bottleneck, is naming the organism. Indeed, we need experts who can handle the Latin names and can get the nomenclature correct. I do not see this as a bottleneck here. We have, for the IGSCM, a very good team of 4 experts who do the work. We are always prepared to accept requests from others who are submitting to other journals to assist them in naming the organisms. And as far as I can see, the nomenclature, quality control, is now in very good shape. My last comment is about the new revision of the ICNP, the International Code of Nomenclature of Prokaryotes. Currently, at this moment, the ICSP is voting on the 2025 revision of the code. The voting procedure will end at the end of October, and I hope that the document can be submitted in the beginning of November. I am very grateful to SJ for having invited me to come to Qingdao, and I will there present the main features of the 2025 revision of the prokaryotic code, and we can further discuss this in Qingdao. Thank you.
Thank you, Aaron. SJ, would you like to reply to what Professor Aaron Oren had said, or would you like to proceed with the question answers?
I think I will respond a little bit about Professor Oren comments. Actually, I give my very deeply thanks to Professor Oren. You helped me a lot with those Latin names and so on. So actually, the name published either validate or not validated. Many of them, I think most, not 100, maybe 90%, you have worked, have helped us. So, or great thanks, thank you very much. This is one thing I first need to, I would like to acknowledge. Uh, yeah, we do have an ambitious aim to discover to find 1,000 new organisms in my lab before I retired. We are working with this these targets. But those are really the work from my lab. The student and the other people, they work a lot yeah, on this. We consider this is very important work to find, to cultivate, not only find. Some people find a sequence, they claim they find a new organism. We really we cultivate the organism and we do some characterization. That's the third and last one, yeah. I'm not here complaining any of those any of those how to say rules or standard for nominates. I just share my experience in my research, my experience to some probably some of those license. For example, publish your description of new organism as a supplementary. Do not do this. Uh, uh, so I share those experience, either good or bad, or some happy, some not happy, with those with people who on the online, particularly young researchers. So they will not repeat my experience. Also, I share my the protocol used in my lab. They are quite. I think they are easier. As Professor Oren said, indeed, there is no certain requirement for what you should do, you must do for phenotypes. You need to find the key features for your organism and describe it. That's yeah, that's it. But this is you know, it's on the document. In practice, you need to find what you will do. Oh, I remember also the publication period, the duration for HSM. I think it's improving. As in that slide I showed, I cited the land flowchart from the land publications. This is really depending on, I think, reviewers and editors and so on. It's quite a lot of things. So I'm not complaining about IDSA. No, not the editors, not the journal. Yeah, I want to make this clear. Yeah. So.
Okay. Thank you, SJ. So we'll proceed with the questions. I have received a few questions. I'll take them based on what is relevant. So the first question has come from Shumaila Nawaz. And the question is, if you have too many isolates that are resistant to a particular antibiotic or something, um, is it required to sequence all of them or just a subset of resistant strains for the identification as a novel taxa?
Okay, what I did, what I'm doing also in my lab is if we find, let's say, 10 isolates, and those 10 isolates can be 10 isolates represent one species. Uh, if you uh, if for antibiotic test is actually I do not understand this part quite. For antibiotic test, for antibiotic resistant test, we just or for sequencing, we just select one or two and maximum three strains to sequence, do the genome sequence, and do the test. We do not do all the same. How to select those one, two, three is we find the it's easy to find the 16S. The 16S, if the 16S is 100%, we just pick up one. If the 16S is 100% identical, but they are from different hosts, and then we also stack the two representatives. So we think probably the 16S is the same, but the other part of the genome is different. And the third is, if you have a 16S difference already, let's say they are 99.5% identity, but still there's quite some difference. There's 0.5 difference. Then you can select both as a representative. So that's what we did, what we are doing. Yeah.
Okay, thank you. The next question has come from Hajra Qayyum. The question is, what about microbes that cannot be isolated and cultured? In this case of metagenome assembled genomes, what would be the pipeline of validating if it is a novel species, given the limitation of the reference database?
The first part of this question, I think, is you cannot cultivate organism, but you
Right,
have the sequence, right? Yeah.
right.
So my belief is every organism can be cultivated. It's about time, experience. So finally, organism can be all microorganisms can be cultivated. Yeah, but currently there are indeed a lot of organisms you cannot cultivate in the lab. That's the fact. For those in my lab, we never claim those new species or new taxa and so on. So as long as it's not cultivated, we will not. There are probably a new gene, a new sequence that's there. There are probably a new organism there. So that's what we what we believe or what we agree. So if you really have a genome assembled from metagenome, and it really represents a new organism, although that organism is not have not been cultivated. I think C code right now they agree that you can give a name of this genome. It's a new organism. as long as the genome, the assembled genome is, I think there is standard of less than 5% pollution of DNA, and also there is other criteria on this. So you can do this if you have really a new genome, but not represent or cultivate bacteria. Yeah.
Okay. All right. The next question comes from Hitisham Haq. The question is, what approaches can be used to validate the characterization of novel taxa in a microbiome research?
The question is, what do you need to validate the?
Yes.
I think Professor Oren online. Um, well, I pass on this to Professor Oren. He's really great on this. Is this okay?
I think that the question is not from the nomenclature perspective, but it is from the experimental perspective. That if you want to check an organism if it is novel, how do you do that in your lab?
Okay. This is my question.
Yeah.
Back to me. So what we do is first we check the sequence. That's easier. As long as you do cultivate organism. Nowadays you have the 16S. Most time you have the whole genome. So we pay attention to culture the strains that have system as identity less than 98% to other known organism. Then we pay attention to those estates. And we do further the genome comparison and do some carbohydrate testing and so on. And then we summarize all those results. We define a new organism or its organism that already been described. So, but anyway, if less than 98%, the 16S identity is a new strain. It's probably an interesting strain in the human gut microbiome project in my lab. We can consider all those strains are important. So probably those, particularly those who are related to health or disease, we characterize those strains. Yeah. Probably not a new strain. Yeah. But
Okay. Yeah. Next question comes from Muhammad Shehzad, and the question is quite generic, but it's important. The question is, what are the major challenges in aligning microbiome research with classical bacterial taxonomy?
So can you just repeat? I need to
Yeah.
digest. I understand every word, but I need
Yeah.
to digest. Yeah.
Yeah. A major challenges in
Mm.
combining microbiome research and classical bacterial taxonomy.
Yeah. This is this is a point that I want to address in my talk actually, and I want to talk a little about this. The major challenge I think is workload. If you do if you do work on microbiome, as I talked, often you met a lot of isolate, a lot of strains, and so workload is really heavy. You need quite some people, a lot of people, human labor, and a lot of experiments. That's one, I think it's a challenge. It's really a challenge. At least in my lab, it's really a challenge. Student work in the lab, student in lab, they really work hard. That's I said all those new microorganisms actually they discovered in my lab. The students and the other my colleagues, they contribute the most. And the second is, I think it's also there is uh, uh, probably some people, not all people, the limitation or the challenge is from the experience, the knowledge you nominate or you claim a new organism, indeed you need to understand the rules from ICSP, also probably the recently the code seek and so on. You need to understand the rules and so on. That's not every people, not all the students, not all yeah, the researchers know this. That's I think a lot of people work in the microbiome part. They even don't know what is ICSP and so on. So that's quite people from medical part field, from medical field. They also do cultivation and so on. Yeah, that's a challenge, I think. So.
Okay. Yeah.
mm.
The next question is again a bit generic, but then the question is, how do you predict microbiome function in health and disease? Do you use any specific tools? And whether bacterial taxonomy is involved in these predictions?
Yeah. Um, yeah, this is probably I did not make clear at the very beginning of my talk. When I gave the title of my talk, I did not think too much, but when I prepared my
Hmm.
slides, I realized probably there are misunderstanding of taxonomy on microbiome. This my talk is
Hmm.
not how to classify microbiome. I know there is good scientists working on this. They distinguish, they classify different microbiomes, some health, some not health, some related to
Hmm.
certain disease and so on. They have really interesting or other words complicated softwares and so on. Uh, uh, those tools they use for classify different microbiomes. I'm not an expert on this and not much experience, but there is quite some literatures on this. So probably can look at those. Yeah.
Yeah. Thank you. Um, I'll take one of the last questions from Mir Sadiq Shah. And then I'll hand it over to Fanas to actually. Fanas, would you like to answer this one, or would you like to go ahead a bit later? You are muted.
I just want to make another comment not related to this one.
Okay. All right. So I'll just take this last question, then you can add. Okay. Um, so the last question that I'm taking is from Mir Sadiq. The question is, how do you expedite predictions of microbial taxonomy using bioinformatic tools in your analysis processes?
So the question is to me.
Yeah. How do you expedite the prediction of microbial taxonomy and functions in your process analysis? Do you use a single computer? Do you use a cluster machine, or what packages do you use? Any information on that that can help our young microbiologists?
Mm, uh, we have in-house let's see. a big computer. What's it? Handling. Probably someone can help me with English. There is called a special name for this. A big computer and also database in that computer. To we input our sequence of data, and then the computer can give some prediction on based on the sequence and the function, so on. But that's not reliable. We always if we are really interested in one organism, we do experiments. Prediction, yeah, prediction is helpful, but right now it's I think not 100%. Although the AI, artificial intelligence, those kind of things really provide good prediction. Still, I think I'm an older man. I believe what I see and what I find. So,
Okay,
yeah.
thank you. Fanis, over to you.
Thank you very much for your talk. It was very interesting. And I fully agree with you that it often happens that we rediscover and rediscover new species, So you isolate it, and by not giving it a name, the next person also comes, and we never gather that information together. And that's why I am one of those promoting the naming of anything possible, even if we don't have a good description, but we have a good thing to compare it with. And I believe that the genome would be the standard to compare it with. So, but I also believe that cultures is important. And if you have a culture, because you cannot make something from sequences, but if you have the culture, you can do lots of experiments with it later. But in India, Brasilia, South Africa, we have the problem that we cannot necessarily deposit in other countries, so we sit with one. And therefore, the comment that I want to make is we should also remember that the C code allow for strains to be named based on their genome. So if you have the strain, it is in a collection, and you have the genome, and you've deposited the raw data, you can name it under the C code. So, and we've done that with isolates because we are restricted in terms of having it validated under the ICSP because of the rules. The other issue is the supplementary data. I know that often you have a large thing, you put it in, you just don't want to just describe the species. You are actually talking about the microbiome, the functions, the things. And just as an aside, you want to name these species. And I think there are also opportunities where one can do that, even if it's in the supplementary with the C code, as long as you have the genome and as long as you have the raw data also submitted to the data banks, then one can have those names. And we need those names because otherwise people will think they've got something new and we're not accumulating the information. So I believe one of the things that we should do in future is we should have a cumulative description, having a name, and with time people add information, we will know the organism better. And if nobody adds to it, then it was something that probably just needed a name, but nobody else is interested, and that's fine. But at least it is there as an anchor. That's all I wanted to say. But thank you very much for your talk.
Yeah, thank you, Fanis. Thank you, SJ. Aaron, any questions or comments?
I posted another comment in reply about naming Max. You can
Yes.
read the comment that I posted.
Yeah, I sent it forward to the person who asked that question. Okay. An excellent discussion followed by an excellent talk. After an excellent talk. So thank you, SJ. Thank you, Fanas. Thank you, Aaron Oren. And thank you, everybody else who joined us, who attended this session. And we look forward to seeing you in the next month. Thank you once again. Thank you, SJ.